Hello everyone. In this lecture, we will be discussing about the saccadic system. As the lecture is quite long, so we intended to separate it into two parts, saccadic system part 1 and part 2. In part 1, we will be knowing the general things about the saccads, the characteristic features of saccads, the functions of saccads, the pulse and step innervation for the saccadic eye movements, and about the neural integrator. In part 2, we will be dealing with the neural substance for the saccads and also the clinical testing for the saccads. The saccads are the fast conjugate eye movements that move both the eyes quickly in the same direction to foveate the object of interest. The stimulus for the saccads can be visual, auditory, tactile, or even memorized targets. Like once you read the book many number of times, you can easily skip from the second line in the first paragraph to the last line in the last paragraph, if you memorize that earlier. And the saccads can be generated reflexively, and they are responsible for resetting the eyes back to the mid-orbital position during vestibulo-ocular or optokinetic stimulation. That is, during the vestibulo-ocular reflex, the eyes might be moving to the eccentric position. The saccads bring them back to the mid-orbital position. Saccads need to be very fast to get the eyes onto the target. And they also need to be very fast because our eyes act like the cameras with the slow shutters. Vision is so blurred during the saccads. So that's why the eyes have to move quickly to minimize the time during which there is no clear image on fovea. Approximately, humans make around 1 lakh saccads every day. So speed of the saccad depends on the size of movement. Coming on to briefly the neural substance, the burst neuron circuits in the brainstem provide the necessary motor signals for the extraocular muscles for the generation of saccads. The burst neurons in the brainstem. Within the brainstem, there is a division of labor between the pons and the midbrain. Whereas pons is engaged for the horizontal saccad generation, the midbrain is engaged for the vertical and torsional saccad generation. The cerebral cortex decides when and where to make saccads. The cerebellum is important for maintaining saccad accuracy and also for adaptation. It regulates the size of the saccads and repair them. From the cortex, which decides when and where the saccad has to be made, particularly the frontal eye fields, the parietal eye fields, the signals directly go to the burst neurons in the brainstem. The signals from the cortex also reaches the superior colliculus. From there, it reaches the burst neuron circuits in the brainstem. That means from the cortex, the signal reaches the burst neuron circuits in two ways. That is, one is the direct way and the second one is indirect way through the relay in the superior colliculus. The frontal cortex, that is the frontal eye fields, also relate to the superior colliculus. The substantia nigra pars reticulata of the basal ganglia within the brainstem, there is a division of labor between the pons and the midbrain. Whereas pons is engaged for the horizontal saccade generation, the midbrain is engaged for the vertical and torsional saccade generation. The cerebral cortex decides when and where to make saccades. The cerebellum is important for maintaining saccade accuracy and also for adaptation. It regulates the size of the saccades and repair them. From the cortex, which decides when and where the saccade has to be made, particularly the frontal eye fields, the parietal eye fields, the signals directly go to the burst neurons in the brainstem. The signals from the cortex also reaches the superior colliculus. From there, it reaches the burst neuron circuits in the brainstem. That means from the cortex, the signal reaches the burst neuron circuits in two ways. That is, one is the direct way and the second one is indirect way through the relay in the superior colliculus. The frontal cortex, that is the frontal eye fields, also relate to the superior colliculus. The substantia nigra pars reticulata of the basal ganglia exert an inhibitory effect on the superior colliculus. The cerebellum is needed for the saccade accuracy and for the adaptation. Here we show the most simplified pathway for the saccade. From the frontal eye fields and the posterior parietal cortex, the signal reaches the superior colliculus, from there to the burst neurons in the brainstem, and from there directly to the oculomotor neurons. The burst neurons for the horizontal saccades lie in the paramedian pontine reticular formation in the pons, and the burst neurons for the vertical and the torsional saccades lie in the rostral interstitial nucleus of the medial longitudinal fasciculus, which lies in the midbrain. Coming on to the characteristic features of the saccades. The first one is regarding saccadic velocity. The saccades can be having the speed of 30 to 700 degrees per second for amplitudes of around 0.5 to 40 degrees. That means if the eye has to move a distance of 0.5 to 40 degrees, the saccadic velocity can range from 30 to 700 degrees per second. If the saccade is to be larger, the peak velocity also will be very high. This is what we call it as the main sequence. The main sequence is an astronomical term where all the stars are graded on a sequence depending upon their temperature and color. So the main sequence is used to determine whether a particular eye movement is a saccade or not, and you also can determine the saccades are abnormally fast or slow. Usually we see the slow saccades in spinocerebellar ataxia type 2. Coming on to the duration of saccades, the duration is linearly related to the amplitude. If the amplitude is more, the duration will be more. So 30 to 100 milliseconds for amplitudes ranging from 0.5 to 40 degrees. Next is the accuracy, saccadic accuracy. So small degrees of conjugate and disconjugate saccadic dysmetrias are normal. Small overshoot, what we call hypermetria of saccards, are common with small amplitude saccards. That means if the eye has to move for a small amplitude, the signal might be little bit higher, so it moves, that is, it crosses the target and moves out. That's what we call hypermetria. Small undershooting, what we call hypometrias, are common with large amplitude saccards. So the degree of dysmetria is normally about 10% of the amplitude of the initial saccade. But it is more prominent with increasing age, fatigue and inattention. So while performing saccade, you need to ask the patient to pay attention, because if the patient is inattentive while you are performing saccades, dysmetrias could happen. So, dysmetric saccades with normal velocity, you can see in spinocerebellar ataxia type 6. Whereas in spinocerebellar ataxia type 2, the velocity will be slow, that is, the slow saccades will be there. Usually hypermetrias are seen in the cerebellar disease. Coming on to the saccadic latency. The latency for the saccade is around 150 to 250 milliseconds. That means the interval between the appearance of a target of interest and the onset of saccade is what we call as latency, saccadic latency. It is about 150 to 250 milliseconds. So the introduction of the brief gap of several hundred milliseconds between the disappearance of the initial fixation target and the appearance of a peripheral target, this is called as the gap paradigm. That is like disappearance of the fixation target and the appearance of the new target. There is a gap. This gap paradigm leads to a reduction in the saccadic latency to 100 milliseconds, where those saccades are called as express saccades. Conversely, if the initial fixation target remains while a saccade is made to the new target, that is what we call as the overlap paradigm. The saccade onset is delayed. It will be around 200 to 250 milliseconds. The gap and the overlap paradigms illustrate the effects of fixation and attention on the initiation of the saccades. So difficulty in initiation of voluntary saccades is usually seen in oculomotor apraxia. So till now we have seen 3 clinical conditions or 4 clinical conditions particularly. Slow saccades in spinocerebellar ataxia type 2, dysmetric saccades with normal velocity spinocerebellar ataxias type 6, saccadic hypermetria cerebellar lesions, and difficulty in initiation of the voluntary saccades in oculomotor apraxia. Now, coming on to the innervational changes of saccades. So for every saccade, there will be 2 components. The first one is the pulse, the second one is the step. The pulse will be high frequency burst of phasic activity in the agonist motor neurons. The pulse is needed, the pulse will be like a throttle or a thrust while you are going up. The amount of thrust what you use on the accelerator while the vehicle is traveling uphill is the, or when the vehicle is you start the vehicle and just move initially. The amount of thrust what you give on the accelerator is called as like akin to the pulse of the saccade. It is needed to overcome the viscous drag in the orbit, and it is responsible for the rapid eye movement. Whereas the step, it is the continuous innervation, holds the eye in its new position against the orbital elastic recoiling forces. So in the eye, to overcome the viscous drag, a pulse signal is needed, and once the eye is moved to the desired position, to maintain it there against the orbital elastic recoiling forces, a step signal is needed. So, the pulse signal directly reaches the agonist muscle. But whereas the step signal is usually a eye position command, and it is created from the pulse signal, which is an eye velocity signal, eye velocity command. The step is a eye position command, whereas the pulse is a eye velocity command. So the step is being derived from the pulse by a neural network that integrates the conjugate eye velocity commands into an appropriate position coded information and relays it to the ocular motor neurons. This neural network is called as the velocity to position neural integrator, or simply the neural integrator. The neural integrator for the horizontal saccades lies in the medulla, particularly the medial vestibular nucleus and the nucleus prepositus hypoglossi. The neural integrator for the vertical and torsional saccades lies in the interstitial nucleus of Cajal. The step signal that is formed by the position command given by the neural integrator contributes for the gaze holding mechanism. So if the neural integrator is failed, so then the gaze holding mechanism is gone. Thereby a gaze evoked nystagmus can appear, whereby the eye is brought from the eccentric position towards the center by the recoiling forces, because the the step signal is not there, and again a corrective quick phase towards the eccentric position, giving rise to gaze evoked nystagmus. So failure of neural integrator leads to formation of a gaze evoked nystagmus. The cell groups of the paramedian cell tracts, that is the PMT, also contribute gaze holding. They relay the eye signals to the vestibulocerebellum. The nucleus paraphilis, or the medullary nucleus paraphilis, which is a part of the paramedian cell tract, receives the eye position signal from the interstitial nucleus of Cajal, and it relays it to the vestibulocerebellum. So any medullary lesion that involves this paramedian cell tract group, particularly this nucleus paraphilis, leads to upbeat nystagmus. Why? Because The nucleus paraphylis receives a position signal from the interstitial nucleus of Cajal for the vertical gaze holding, and if this nucleus paraphylis there is a lesion over there, you can expect an upbeat nystagmus. And also there is another point here, that is in the nucleus paraphylis lesions, which also involves the lesion of the ventral tegmental tract pathway for the upward v4, which is mediated by the superior canal, leads to a downward vestibular bias, and the eye will be pulled down, thereby giving rise to upbeat nystagmus. This pulse and step innervation applies to all types of eye movements. In smooth pursuit movements, the vestibular movements and optokinetic slow phase and vergence, the phasic increase is smaller than that required for the saccades. So, any saccadic disorder can be a result of an abnormal pulse or abnormal step or a mismatch between pulse and step. So in cases of leaky neural integrator, the pulse will be normal, the step is unsustained. So the eye drifts towards the central position at the end of each saccade. That's what we call it as gaze evoke nystagmus. So, in the part 2 of the saccades, we will be discussing about the neural substance for the saccades and the clinical examination of the saccades. So, that brings us to the end of this topic, part 1 of saccades. Thank you.